Yo, yo, yo. So this is a quick overview of the biomolecules. Don't use this as a way to try to memorize it or think that this is all of the information that you will need for your exam. It is, again, to frame your thinking. So we're going to start off with carbohydrates. So carbohydrates. And again, we always need to know the elements that makes up this biomolecule, and we will remember it as CHO. But there's another CHO, so we need to know the difference. So this is in a ratio. And if we have certain number of carbons, we have to multiply the hydrogen and oxygen accordingly. And again, the most common one would be C6H12O64 glucose. Okay, so speaking of glucose, we need to remember the information in terms of monomer, polymer, the functions, and then the bond that makes it up. So we're going to start off with monomer. So with the monomer, it is considered to be the monosaccharide. And with the monosaccharide, we know it's just one subunit that could link up with others to create the polymer, right? But before we get onto that, I'm going to give you guys an example. And again, it is glucose. And we need to know the function. So the function for glucose is quick energy. Okay. And then we also have that linking. So monomers link up together to form these things called polymers. And so what links them together? Let's do that real quick. The bond for carbohydrate, the covalent bond, is glycosidic linkage. And then for polymers, it is a polysaccharide. So many monosaccharides link up by the glycosidic linkage will create polysaccharide. For the polysaccharide, it's very cool that we can categorize the four we need to know specifically for AP Biome by their function. So function will be here. And number one, we have energy storage. Okay, energy storage. The first one we have would be glycogen. Again, animals. And we have starch, which is plants. The second function would be structural support. Okay, the structural support includes cellulose. And for cellulose, cellulose is the component of the cell wall for plants. Then we have chitin. Chitin compose the exoskeleton for a lot of animals. Okay, so including crustaceans and um, insects. And then we have them also as the cell wall for fungi. So this is not we also need to know that these polysaccharides are made up of those components, and those components dictate that structure. So we're going to highlight two big differences: starch and cellulose. So they're monomers, they're both glucose, but two types of glucose. So for the starch versus cellulose, we're gonna need to note that it is the alpha glucose making a very specific bond. We don't need to know that specifically, but we need to know that starch is, helical, and then there's some branches that branch off, and this helical structure allows for enzymes to come in and to hydrolyze the glucose monomers apart for a quick source of energy. And then we have the cellulose on the other hand. Again, basically due to their type of monomer and how they link up. They create a very specific shape. So this one is the beta glucose, and their shape is a very linear line. And in between it, hydrogen bonds can form, strengthening the structure. Thus, we know that is there for a support. And again, that's all you need to know for carbohydrates. Again, remember, go back to your notes to relearn any of the tiny details that's not here. Anything in the study guide is fair game. And then we have our second category, and I'm gonna be writing this in, let's go with red. We have the lipids. Okay, lipids are special. We can't think of them in terms of their monomer because they don't have true monomers. The parts are different. They're more categorized by their properties. So let's go over the component. So it's CHO, but here's the key difference, y'all. With this CHO, there will be less oxygen. Okay, that's going to be the big difference between CHO for lipids and CHO for carbohydrates. CHO for carbohydrates is in a specific ratio, a 1 to 2 to 1. There's no such ratios here. There's usually a long chain of carbon-hydrogen bonds, and these are very high energy, and we're going to talk about that soon. And now we need to note that they are group again due to their non-polar characteristics, okay, or hydrophobic characteristics. Now, we need to know three special groups. The first one would be fats, and another name for the fats would be triglyceride. Okay, for this, we still need to know what makes up a triglyceride. So the components or parts would be glycerol and three fatty acids. And the structure will look something like this. Their structure, again, is good for long-term energy storage. And another function of fat would be a great insulator of heat. And so it regulates that body temperature well. Okay, for fats again has a special category. We need to know the difference between saturated and unsaturated. So for saturated fats, we have the 3S mnemonic device. So again, saturated for single bonds, and this is specifically the single carbon to carbon bond. And then we have them being solid at room temperature due to their structure. Their structure is a straight line that you guys can see here. They can stack on top of each other, so forming a solid. So the 3S. For the other one, if you remember the 3S, you'll automatically know the opposite, which is unsaturated. So unsaturated has at least one double carbon-carbon bond, and this creates a bend or kink in their structure. And that kink doesn't allow for the lipid to stack neatly on top of each other, so they remain liquid at room temperature. So let's draw an example of that real quick, and we can see that they cannot neatly stack on top of each other if there are multiple of these unsaturated fatty acids. So let's go backwards and concentrate on that term saturated versus unsaturated. Saturated refers to the maximum amount of hydrogen bonds that can be bonded around carbon. Okay, so saturated actually means it's full of hydrogen. And the unsaturated is not full of hydrogen. And then, yeah, so let's look at our second category. Our second category would be phospholipids. Okay, phospholipids. Phospholipids, the parts, includes, and I always go in this order, in order of the structure, a phosphate, glycerol, and then two fatty acids. I haven't mentioned the bond that links these guys together, and it's usually the bond between a glycerol and fatty acid. So you need to know that the bond's name is called an ester linkage. It links the glycerol and fatty acids. So ester linkage. It's a bond that's a COC bond. Okay, back to the structure. We're going to draw the same thing. It has something like this. This region here is considered to be polar or hydrophilic. And then we have this part to be nonpolar or hydrophobic. So again, this phospholipid makes up the cell membrane. Cell membrane is made up of a bilayer, okay, lipid bilayer. It looks something like this, where the phosphate head is facing the aqueous or liquid water facing area, where the tails, the fatty acid tails, are turned inwards away from water. Okay, and then what's the purpose of that cell membrane? So it has some functions, and let's just go over the functions so you know. It again protects the cell from environment. And then we have the second function of allowing small molecules in and out of the cell. So again, cell membrane, there we go. Now, we have a third category, and it's called a steroid. Okay, the steroid is, again, 4 fused rings. That's nonpolar. So let's draw the structure like we saw before with the others. It looks something like this. And then we have our, oh, mess up there. It's like this. Okay, we have this, and this is nonpolar. The big example we need to know is cholesterol. Cholesterol, as we learned in class, has a polar portion that can attach to the cell membrane to affect its membrane fluidity. And so this right here should be an OH. Let me clean it up. OH. And then the rest of it, there's a long chain as well there that's nonpolar, a hydrocarbon chain. And that part would stick to the fatty acid of another phospholipid. So cholesterol affects animals cell membrane. Okay, that's what you need to know for the lipids. Again, go back to your notes to make sure that you're covering everything in the study guide. Let's dive into proteins. Again, proteins are my favorite. Let's go. Proteins are made of chons. Yes, we added the S, and the N is something new from the previous categories, right? Again, we can remember the information with the monomer, polymer, and bond. The functions, again, you guys should just look in your study guide. They follow the English language, like transport proteins, again, allow things in and out of the cell. It transport, right, things, so it's common sense. Defense, again, very easy. Y'all can go over that stuff on your own. But let's go over the nitty-gritty details of monomers. Okay. We again have the amino acid. Amino acid. And it's good to know the structure. You don't have to memorize all 20 different amino acids, but to know that it has an amino group, an R group, especially the R group, and that new amino acid are added to the carboxyl end. So carboxyl and amino. So carboxyl N, new amino acids are added. And, you know, this should help y'all out as I go through the structures of amino acids and the structures of the protein. So let's go. We have the polymer. The polymer has two names. Again, we have the polypeptide and proteins. Okay. The bond, and this is crucial, that links those amino acids together is a covalent bond, just like all the others, created by dehydration synthesis, the removal of water. And then we see that it's called the peptide bond. If you see the word peptide, hint, hint, it's gonna be a protein. Whoa, 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 not that bad. Okay. We have what we need now, so let's dive into the structure. So there are four structures y'all need to know well. So I'm going to start here. Four structures, okay, of protein. The first one is the primary structure. The primary structure can be defined by a sequence of amino acids. Okay, I like to think of it as a chain, and I like to depict these guys in drawings. So again, just here you can see the peptide bond, and we need to know the bonds for each level. So the bond you can see here that determines this structure is just the peptide bond, linking the amino acids together. Okay, so for number two, I'm going to move my pen over here, and we have the secondary structure. Secondary structure comes about when the amino acid chain starts to twist and turn. Okay, so this twisting and turning creates two shapes, the alpha helix and beta pleated sheets. Very specific structures. I'm going to draw the best I can, and I'm not that great at drawing, so bear with me. And then we have the beta pleated sheets here. They're all held together by this very specific bond, and it's the only one that determines the shape here. Okay, so bond would be hydrogen bond, and it's again between the polypeptide backbone. I repeat, the hydrogen bonds between the polypeptide peptide backbone. That backbone includes everything you see here for an amino acid. And the big thing is that there's no R group interactions at the second level. That's crucial, okay? For the second level, no R group interactions. Now, as again, the R groups come into play, we have our third structure. It's called the tertiary. Yes, you guessed it. It's going to go in order. Tertiary structure. So this tertiary structure, I like to call it as a globular shape. I'm going to draw it for you too, like something like this. It twists and turns, bends and folds, and does all the things you would see a protein be. And it's because of the R group. So due to R group interactions. So again, we need to know the very specific interactions, but before that, let's define the properties of the R groups found here in the 20 amino acids. Like I said, you don't have to know the 20 different amino acids, but you need to be able to categorize them in groups and then describe their bonds. So there are 4 groups you need to know. Again, hydrophobic, and that's non-polar. Hydrophilic, polar. Remember, polar is the partial positive, partial negative of a molecule. And then we have positive and negative charge, very different from the polar. Okay, this is totally positive and this is totally negative. Now, these 4 different properties, again, create the 5 interactions you need to know. So I'm going to write it over here, interactions. So the interactions, number 1, hydrogen bonds. So again, between a polar and a polar group, we would have partial positive on a hydrogen. Okay, so this is a hydrogen bond. You need to know what it means. And it can bond to either an F, O, or N that has a partial negative charge. The partial attraction of the opposite charges creates that hydrogen bond you see. Our second one would be the disulfide bridge. It's a cross-link between 2 sulfur atoms in the R group. And so again, let's not forget that it is a covalent. Okay, so cross-linked, and covalent. Between 2 S's. Okay, so S, S. We have our third, hydrophobic interactions. So with hydrophobic interactions, it just means that nonpolar is going to attract nonpolar substances. Nonpolar attracts nonpolar. Remember, the nonpolar would be those hydrocarbon chains. That's not charged at all. Number 4, we have our van der Waals. So van der Waals is considered to be just weak interactions. That's all you need to know, weak interactions. The interactions come about because electrons are always moving. When the electrons are moving away from a region, it creates a positive, temporary positive charge. And when the electrons are moving towards a region, it has that temporary negative charge. And that temporary charge interaction is considered to be van der Waals. But if that was too complicated, just think about it as weak interactions. And then number 5, we have ionic. ionic is not between polar. Okay, polar has the partial negative, partial positive. This is between a positive charge and a negative charge attraction. Okay, so ionic. Now, usually speaking, the tertiary structure is the final form of the protein, and only certain types of proteins have this last structure here, which is considered to be the quaternary structure. With quaternary structures, we know that it is two or more tertiary interacting to perform one function. So this example is very specific, hemoglobin. That's usually the go-to bio example for a quaternary. But remember, usually this right here is our final form of a protein that has a function due to its shape. And then over here, y'all, remember, no function for the primary structure. Okay. This is a very, very quick overview. Again, more details in your notes and study guide. Make sure you look over that. Sticking with the Spartan colors, we have nucleic acid. For our last biomolecule, again, it's made up of CHONP. So the new one is phosphorus. Okay. Now, again, easy to remember. We need to know monomer, polymer, and then the bond. Monomer would be the nucleotide. And the nucleotide actually has parts to it that you need to know. 3 parts to it. So the first one, again, I like to do it in how it's structured. Phosphate. Number 2, we have the 5-carbon sugar. And finally, number 3, we have the nitrogenous base. Okay, so with that said, we have this right here. You've seen it in the notes, you've seen it in your stations. So let's label them real quick. 1, 2, and 3. Nucleotides. Everything you need to know right there. Let's dive into the nitrogenous base real quick. So I use one mnemonic device to remember both categories. So we have cut, pi. So what does that mean? Pyrimidine, right? Stands for that pi, pyrimidine. And you just need to know that it is 1. These bases, CUT, has 1 word. So 1 ring. 1 word equals 1 ring. And so that means that pyrimidine is a 1-ringed nitrogenous base. So by default, the other ones would be 2 rings. So let's list it out. The other ones are called purines. And if it's not CUT, obviously going to be AG. Okay. And this is going to be 2-ring structure. And that's it. 2-ring structure. Boom. Easy. Now, we have the bond that links all of these guys together, right? This bond has a complicated name. It is the phosphodiester linkage or bond. This right here, this covalent bond is only between the sugar phosphate backbone of DNA or RNA. Okay. Very crucial, not with the nitrogenous bases at all. So we have the polymer. It could be called a polynucleotide, very uncommon, or DNA, or RNA. Okay. So let's draw out the process of creating that phosphodiester linkage, and then we can note other things as well that you would need to know. Let's draw it out. So we have this here. Again, very bad at drawing, so bear with me. Again, link here. So DNA, similar to the protein, needs to be added in a certain direction. So on the 5' end, we have a phosphate group. So again, phosphate is always going to be 5' end. On the other end, which is not listed here, and it rarely is the 3' end with the OH, the hydroxyl group. Okay, so new nucleotides are added in the 3' end. So what create these bonds? Let's review that because you do need to note. So that's the phosphodiester. So again, in order to build that bond, we need to dehydrate it, right? And then to break the bond, we need to add water. So adding water to that bond, again, means hydrolysis. We're going to break that covalent bond. Breaks covalent bond. And then on the other hand, if we want to form that bond, removing water, this is dehydration synthesis. Again, this is true for all the other polymers, and then they're all covalent bonds. Okay, so dehydration synthesis. And we need to know one last thing. This runs from 5' to 3'. So for anti-parallel, when these polynucleotides are linking up, there's a second strand. This side would be the 5' end, and on the other end would be the 3' end. How do they link up? Well, the nitrogenous base right here links up through hydrogen bonds with the other complementary nitrogenous base. So A pairs with T, and I draw the two lines to represent two hydrogen bonds, and then C pairs with G, again, three hydrogen bonds. And that's all y'all need to know. Again, go back to your study guide, but this is a big overview. Have a good one.